ICI 180-780. But from previous studies in our lab, we know that our hypothalamic astrocytes do not respond to 17-alpha estradiol. And I kind of showed you earlier that ICI 180-780 actually can block the estrogen response. So,
John, very, very good point that they use 17-alpha estradiol here. I'm just being the devil's advocate. As 17-alpha estradiol can produce some actions, and probably most likely due to maybe its antioxidant action. One, I think the good part is you blocked your things with the ICI, and 17-alpha was not blocked by ICI.
well, for these receptors, it's not blocked, right? And then, so we don't think that it's ERX, and we actually eliminated this because one, ICI 182-780 blocks in our hypothalamic astrocytes, and 17-alpha doesn't activate. So we automatically ruled the last one out. So the other four we're actually going to study further. And so the first thing we did is a dose-response curve with several different agonists. PPT is a selective estrogen receptor alpha agonist, and it has preferential binding to estrogen receptor alpha over beta about 410-fold. So these are not pure agonists. And then DPN is a selective estrogen receptor beta agonist, and it has preferential binding of estrogen receptor beta about 70 times higher than estrogen receptor alpha. Then you have STX, which is the selective estrogen receptor modulator that activates the STX binding protein. And then you have G1, which is a GPR30 agonist. So we wanted to see how the different agonists respond in the hypothalamic astrocytes to see which one is the closest to what estradiol does. So in the yellow is the dose-response curve that I showed you earlier of estradiol with an effective concentration, a half maximal effective concentration of 0.15 nanomolars. And so with PPT, which is a estrogen receptor alpha agonist, we see that the response is here. The maximum response is very similar, but at lower concentrations, it was a little bit more potent. And so the EC50 was a little bit lower at 0.03 nanomolars. Then we looked at DPN, which is estrogen receptor beta agonist. And we see that it's actually a much weaker agonist, and it requires about 100 times higher concentration in order to get the response going. But the maximum response is very similar, but it's much weaker. And then STX is in the bluish gray here. So very similar response, but at the higher concentrations is actually a little bit more effective. And then there's G1, which is a GPR30 agonist. And again, that's very similar to the DPN, where it required much higher concentrations for their similar effect. So I just wanted to point out that DPN and G1 are not very similar to, the response is somewhat different than what estradiol looks like. So physiologically, it looks a little different, and it required much higher concentrations. So therefore, for the progesterone assays, for DPN and G1, we actually use 100 nanomolars, as opposed to 1 nanomolar for estradiol, PPT, and STX. So I just wanted to show the rationale for using different concentrations. In our progesterone study, we wanted to see that these agonists will produce progesterone. And so with estradiol, we got a good progesterone response, and we've seen that also with PPT and STX and also G1. But DPN, even at 100 nanomolars, did not stimulate progesterone production. So this tells us that it's probably not signaling through estrogen receptor beta. And then we know it signals through a glutamate receptor, and so therefore we block the mGluR1A with LY36738-5, and then we wanted to see if this would block the different signaling pathways. So estradiol again was blocked. PPT was also blocked, which is estrogen receptor alpha. The STX was also blocked. DPN did not respond to did not respond with the progesterone synthesis. But what's interesting is that the G1 could stimulate progesterone production, but actually was not blocked by LY36735. So this tells us that there's probably a different pathway or some different signaling mechanism for GPR30 as opposed to estrogen receptor alpha or estradiol. So for our calcium studies, we wanted to repeat the same thing and see if LY36735 would block. And actually for all four, PPT, DPN, STX, and G1, here G1 actually, it actually blocked the calcium response, but not the progesterone synthesis. And then we wanted to see what would happen if we added the DHPG in addition, this is DHPG at 100 nanomolars in addition to the different agonists. And we see that there was an increased response in PPT, DPN, STX, but G1 actually did not increase. So again, this is a little bit different than the other agonists. So So what does this mean? So summary at this point, PPT and STX are the most closely mimicking estradiole's effects, which suggests that the estradiol may signal either through membrane-associated estrogen receptor alpha or an STX binding protein. DPN failed to stimulate progesterone synthesis and required high doses for calcium release. Thus, estradiol is not signaling through membrane estrogen receptor beta. And then GPR30 probably signals through its own unique pathway, which we'll talk about a little further. So the next step was to do some co-immunoprecipitation. And in these studies, what we want to do is to see if two proteins interact with each other. So we would lyse cells, and then we could either study the whole cell or just the plasma membrane fraction. And then we would add an antibody to that lysate or fraction, run it on a column that recognizes the antibody so that you can isolate the protein of interest. And then you can elute the column and then run it on a Western blot, and then on the Western blot you probe it with a second antibody. So if the first protein and the second protein are attached, it'll stay on in the column, and now it'll be on the Western. And so when we probe with estrogen receptor alpha, so the first one was mGluR1A, and then after we run on the Western, we try to see if it's attached to estrogen receptor alpha. And we see that there is some association in the whole cell, but it's mostly the association on the plasma membrane fraction, which makes sense because the mGluR1A is a membrane receptor. Then when we looked at estrogen receptor beta, we actually did not see any interaction in the whole cell or on the plasma membrane, which again probably specifies that the glutamate receptor and the estrogen receptor beta are probably not interacting. This is probably not the main mechanism. What was interesting is that when we did this co-immunoprecipitation for mGluR and GPR30, down here, we actually did not see any interaction, which again signals that it probably signals through a different pathway. But then when we looked at GPR30 in just a regular Western blot, we could find it in the whole cell, in intracellular, but we can't find it in the plasma membrane. So this is kind of consistent with other literature where nobody has actually been able to show native GPR30 on the plasma membrane. They've only been able to show it on the smooth endoplasmic reticulum and the Golgi apparatus. So again, GPR30 may be a little bit different signaling mechanism that actually may involve intracellular signaling. So summary of findings: estrogen receptor alpha, but not estrogen receptor beta or GPR30 interacts with the metabotropic glutamate receptor 1A, as demonstrated by co-immunoprecipitation. GPR30 was not identified on the plasma membrane. And so our hypothesis for G1 for the GPR30 signaling is that since G1 stimulated progesterone synthesis was not attenuated by LY367385, it suggests that the G1 may directly activate an intracellular GPR30, such as on the smooth endoplasmic reticulum, to directly release the calcium, which can then stimulate progesterone production. Now, G1 required high doses for calcium release, which may make more sense because it has to go into the cell to signal. But then again, from our calcium studies, the LY367385 blocked G1's effect, but it was not enhanced by DHPG, so that also just suggests that instead of acting on a GPR30, which goes through mGluR1A, it actually may also activate mGluR1A. And so the G1 and the DHPG would act on the same membrane and compete against each other. You wouldn't get a higher effect. But in that, calcium can actually be blocked.
What's the chemical structure of G1? Can it cross the membrane? I mean, is it lipid-filled?
Smooth.
Yeah. Yeah, I can't remember the actual structure. Sorry. So the next step was to basically try to study the estrogen receptor alpha and the STX, which are the two receptors that we kind of narrowed down.